How did you have the guts to jump into a little known startup?
One day I went out to dinner with my dad and he goes, "What are you doing with your life?"
It's no longer about accessing just the genome, right?
I'll kind of nerd out a little bit. I think there's a relationship between these nanowells, and it really was a Eureka moment.
A little bit surprised we haven't actually talked about the box. Hi, I'm Ashley, and I'm so excited to welcome you to Illumina's first video podcast series, The Sequence, where we bring you candid conversations with the people leading the genomics revolution. I'm here in Illumina's beautiful Customer Engagement Center in sunny San Diego, where I'm thrilled to invite our first guest, our Chief Technology Officer and SVP of R&D, Steve Barnard. Steve, welcome to The Sequence.
Thank you. It's absolutely a pleasure to be here kicking off this first series of the podcast with being able to talk about my passions, Illumina, technology, the science, the future. It's going to be fun.
It's going to be so fun. And I'm so excited that you agreed to be our first guest because I know a little known fact about you. And we'll get into your background. But could you just share for the audience what, in fact, your employee number is?
Yeah, so my employee is number 4. I was the first scientist hired at Illumina. And there was really, when we started the company, about 8 key people from the very beginning.
I'm going to have to date you a little bit, but I do know that that was about 3 decades ago. How did you have the guts to jump into a little known startup, employee number 4, before genomics was even a thing, right? Like, what was the road that led you here?
Yeah, it really goes back down to, I think the story is, I was doing medical research in Boston. And one day I went out to dinner with my dad and he goes, like, "What are you doing with your life? You need to get a graduate degree if you're going to stay in science." And that was kind of an epiphany for me. It's time to start moving on, Steve. You're not getting any younger. And I started looking around for places to study in Boston. And I found David Walt Lab. So I went and interviewed with David and just really liked him and what he was doing in his lab. And David was, from my point of view, just an incredible mentor. He had this beautiful combination of creativity, innovation, and understanding of what the real world needed. And that really struck a chord with me. So I joined his lab and turned out I'm pretty good at research. And so I felt I was very successful there. And in my PhD, that led to a number of patents and a number of, Nature and Science publications. So that was a very productive time of life for me. From there, I got a job and I was transferred to Switzerland for 5 years, and I was working at a big pharma company called Ciba-Geigy. So in 96, I moved back to Boston to the subsidiary. David called me up and he said, "Hey, you know those patents you got during your PhD? A venture capital office wants to license some of them. Are you interested in joining a startup?" And I said, yes. It's always been on my bucket list. And to be honest, I had no idea what that meant.
Yeah.
I was very naive, youngster in science, still at the lab bench at the back then. But I said, yes, it sounds really cool. So I loaded up my Jeep and I drove from Boston to San Diego. And probably a fact most people don't know is we started the company in a 2-office building venture group up in a little village north of San Diego called Cardiff by the Sea. And there was one conference room and it was beautiful. It was probably an 8x10 size conference room, but it had this beautiful ocean view. And all day long, we would sit there and watch the waves roll in. And I bring that up because over almost 3 decades, being in Illumina has been riding almost this perfect wave in terms of technology, in terms of influence, in terms of this impact we're having on society. So from my point of view, this almost 3 decades has seemed extremely fast and short, right? So it's actually been a wild ride.
That's amazing. Well, and very apropos for a San Diego-based company, right? Chasing endless summer. And riding those waves. But I love that analogy because I think it gives us, it helps me understand how you've thought about approaching innovation, which is something I really want to talk about with you. because the industry and the market and our customers have changed so much over those last 3 decades, right? I mean, the problems, I would love to hear your perspective on some of the problems that we were solving at the very beginning. And then we'll talk about some of the exciting problems we're working on today. So what were some of those early problems?
Yeah, it's a really good question, Ashley, because if you look back, and that's the privilege or advantage I have of being with the company so long, when we look back to when we started the company, there really wasn't a next generation sequencing field because there was no technology out there doing that. It was mainly centered around SNP genotyping and gene expression technology. So the first sort of wave of innovation Illumina had and was developing the bead array technologies. Those were our first products. Those are the products that got us to profitability. Then in 2007, we kind of read what was going on in the market, and SNP genotyping, gene expression just wasn't going to be enough. And there were emerging technologies like the Solexa technology that was promising the ability to sequence. Okay, now put that in framework, in a little in perspective. The human genome had been sequenced at that point, so there was a reference genome. So what those technologies were really doing is developing technologies to resequence based on the reference. And that's something, a turning point that I think people who don't really understand the decades of evolution of the field really appreciate. So that was a huge turning point. And it really was the second S-curve of innovation Illumina went through was the acquisition of Solexa and the sequencing the SBS sequencing technology. So it's continuing. Right now, today, we're on our third S-shaped curve of innovation with some really exciting products that are coming to the marketplace. So
that's great. Well, and so for those listeners who might be less familiar, let's talk about, maybe these are those waves, right? That we were talking about surfing. So the first wave was the bead array technology. And what was the limitation, I would say, of that technology that set us up for the second wave, which was human genome resequencing largely?
Yeah, so there was a limitation there in SNP genotyping where, the SNPs had to be known. So that technology isn't a known a priori type technology. So you could look at those SNPs that were known and start interrogating them at the global level, so across disease cohorts or populations. But that only kind of increased our knowledge base of genomics so far. When sequencing technology came into play, now you could interrogate the sequence of disease state or healthy state without relying on that a priori technology. You had the reference genome to do that. So now it became the big challenge of how you could develop technology and tools to do that at scale, to do it faster, to do it cheaper, to really bring this unique technology to the market in a large way. So we talk about democratizing technologies, and that's a big question when you think about technology development and innovation at a higher level.
Mm-hmm. Well, and that access and democratization, I think, comes from a couple different vectors, right? So the obvious vector that everybody wants to talk about is cost, right? And so I was in the genomics game at that time, and I remember getting a grant for $1 million, and we could sequence 3 genomes, right? We could sequence a trio,
yep.
Mother, father, and baby for $1 million, about $300,000 a genome. Can you just, catch us up to, with Illumina's core SBS chemistry and technology, what did that change in cost look like during that second wave?
Yeah, it was incredible. And what you're pointing out here is this natural catch-22 of how a market develops, whether it's a commercial market or a science market or a healthcare market, where until the correct tool is brought to the industry or the application space, you don't unlock its potential. And that was the very thing Illumina has done repeatedly. It did that with the HapMap project and with the SNP genotyping costs between 1998 when we started the company through the early 2000s. But then as we developed the Solexa SBS technology, the same phenomena happened. With investment and technology roadmaps, we unlock these price points probably every 5 years that drop that cost, where if you think about the absurd costs when you were doing research, a trio for $1 million, now the price of a genome is hovering between $100 and $200, right? And that's for a plain genome, right? So that's incredible. The secret to all of this at Illumina has been developing a 10-year roadmaps of capabilities. We do that every year through a process called future sequencing. It's a strategic technology planning process. And one thing we did very early in the company was plan a density roadmap and how that density roadmap evolves over the next decade. Now, those roadmaps get created and edited each year. And what we have today is an incredible roadmap where our flagship product, the NovaSeq X, is delivering 16 terabytes of data, right? Which is just amazing to think about at a genome cost, varying between $100 and $200 in general, right? So it's just an amazing inflection point in the industry.
I completely agree, and I'll never forget the first time I got to sit in on Futures of Sequencing, when I got behind the curtain of Illumina after having been a customer for so long in the ecosystem and a partner as well. I mean, my mind is blown. I was literally jaw on the floor seeing that process and what was going to be possible 10 years from then. And I'm also really proud to say that, much of it's come true, right? And bringing those innovations to customers, which is, it's such an honor to be able to do that. So let's maybe talk about this next third wave, right? Maybe it's coming, maybe we're paddling for it. I don't know where we're at, but I would love your take on that. It's no longer about accessing just the genome, right? I mean, I think it sounds like it's much more about how do you get the most out of that, right? So you've made it accessible. Now let's really interrogate what's there. And we just recently announced TruPath as one of the tools in the toolbox to do that. Can you describe what TruPath is? Yeah,
going back to the wave analogy, we're just catching the wave right now. These innovations are so powerful, and we'll talk about a couple of them probably in a couple minutes. But TruPath specifically. So there are regions of the genome that have clinical relevance, and they're so complex that sequencing technologies, whether they're native long read or classic short read technologies, just couldn't sequence through or map back to the reference. TruPath is just an inflection point where I've unlocked that ability to be able to do that now. And it's extremely cool technology. And so I'll kind of nerd out a little bit here, but you can imagine taking non-fragmented DNA. You can picture it like a long spaghetti strand, right? And then taking that and throwing it across the surface of our flow cell, which is an ordered pattern surface. So each of those patterns has information in it and capability. So that piece of spaghetti lays across each of those nanowells, as we call them internally. Our transposase technology captures a part of that long unfragmented piece of DNA and clusters it. But it doesn't stop there. What's amazing here is what we found was that you can associate one nanowell to the next nanowell coming from the same single molecule of DNA. And that was the epiphany that we had. So what the utility of that is now you can link all those nanowells and those informations from the clustered nanowell together. So we're getting megabase size N50s, which allows us to actually interrogate expanded information, large structural variants, being able to understand and map, triplet repeat expansions, things like that. Things we could, a decade ago, we never imagined doing this with our technology. And there's an interesting story about this. Yeah. And this is a good lesson for anyone running R&D. About 14 years ago, we created some IP. We had a group working on how to simplify library prep. So the knee-jerk reaction was, well, we'll do library prep on the surface of a flow cell. And the team got so far, and we looked at sort of the commercial sort of viability of that technology. We said, well, maybe it's not the right time to do that. So we put that technology on the shelf. But about 4 years ago, we said, well, maybe it's time is right to actually bring that project off the shelf, because people are now interested in simplicity. The market's grown to reduced hands-on time. So we started that project. But this time, we put the team together very differently. It was a diverse set of talent. And the new talent we brought into that program from the very beginning was a bunch of bioinformatics guys. So as the project developed, we started creating all this data. And then one day, one of our bioinformatics guys said, hey, wait a minute. I think there's a relationship between these nanowells, the sequencing information. And that led to this Eureka moment of there's a lot more information in those clusters and how spatially they're related to each other. Mm-hmm. And what does it mean for mapping back to the reference genome? And it really was a Eureka moment. So this is the new standard, I think, of whole genome sequencing. You're getting all the short read accuracy, but all the expanded information that was typically only available in native long read technology. So it really is the best of both worlds.
That's incredible. So for somebody who's maybe not as familiar with our core products or sequencing, just by comparison, can you describe the process that somebody would typically have to do to go from, like a blood tube to a sample on a sequencer and then data out? And again, like the simplicity of the information versus what does TruPath do?
Yeah, so TruPath basically eliminates most of those steps up front. And there's probably 3 or 4 steps where you have to take your blood tube, isolate the DNA, and that DNA has to go through some library prep steps, which is about 3 or 4 hours invested in that.
Okay.
And typically you're doing it by hand, or you have to automate it because there's so many steps. TruPath is amazing. Once you isolate the unfragmented DNA, that just loads directly on the flow cell. So it's about 10 or 15 minutes hands-on time, and you press a button, you walk away, and the sequencer's off sequencing. So everything is done on the instrument itself. So we really think this is going to be the de facto clinical genome in many use cases right now. As we start multiplexing this, I really think it's going to move into different market segments as the de facto genome for scaled up many sample type cohorts.
I mean, I wouldn't be surprised. And I also heard a rumor that maybe our CEO was one of your first alpha testers.
Yeah.
Is that true?
So the joke is, if before you say it's simple, you have to ask the C-level to go track to do it in the lab.
Exactly.
So Jacob's done it. I've done it. Eric Green has done it. And it's pretty amazing. People don't really believe us when we tell them, no, there's very little hands-on time and everything is done on the flow cell. But once you do it, you realize the power there. So, yeah.
And it further gets to that accessibility point, right? So it's not just cost, but it's how sophisticated does your lab have to be to be able to process that sample and get it on the instrument. And then ensuring that you're getting the most information out from the genome every time you're running it, which I think is what you're talking about, those harder to sequence regions where they have repetitive A's, T's, G's, and C's, where you can't always count them cleanly. This is the technology to do that. Am I understanding that right?
Yes, it's been amazing. As we've gone through the development program, we started working with key opinion leaders. And the first thing they do is send us the most difficult samples that they're having troubles with. Hemophilia was one disease from a colleague at Rady's Children's Hospital. He said, well, you're not going to be able to do these. I just don't believe that. He sent us 10 samples, and we nailed those 10 samples with the information that doctor needs to actually diagnose the severity of hemophilia. The severity of hemophilia with the F8 and F9 gene, depending on the complexity of the variants, will determine the severity of the hemophilia condition. And using TruPath technology, he was able to classify all those variants and the samples. So he basically said, this is it. This is what I'm going to use this is the technology I'm going to use in the future.
The technology that will underpin
Yeah.
their diagnostics. That is fantastic. So TruPath sounds like it's a really transformational technology. Beyond the hemophilia case, can you give me ideas about other ways that this will help, unlock different avenues of research, similar to, you described how the bead array really unlocked scaling up the HapMap study globally. What will this enable?
Yeah, so based on sort of the early adoption, what we're seeing is that people really want to start embracing it for rare disease. So the interest has just been more than we imagined. And the reason for that is the price point, number one, the comprehensive information that you're getting from it, where previously people would do sort of a short read sequencing run. And then if they couldn't essentially understand the condition, they would do a native long read. So you have this reflection to more complicated technologies. What we're finding is you don't need to do that anymore. How do we get access? How do we start doing the pilot studies to get it into our healthcare system? So it touches on the point we talked about previously. If you enable the technology to satisfy the market or the customer needs, that unlocks this catch-22 that you're in, because now you can do what you want to do, whether it's in healthcare or whether it's in science, to kind of push to the next limits of what you're trying to achieve. So this is really groundbreaking technology. And I know I'm probably overemphasizing this, but when I talk about that third wave of innovation, we're at the very beginning of this wave. The limits that we're going to be pushing this technology to is we just don't know where it's going to end yet. So we're in that phase of exploration. So it's going to be a beautiful story to continue.
I think that is so exciting, right? And it really is that co-exploration with the researchers, with the clinicians who are adopting this technology and then coming back and finding new ways to apply it, right? And I think I completely agree with you. I was just thinking back years and years ago, I had the pleasure of being a judge at the Guinness Book of World Records first run for the world's fastest genome sequence, done by Stephen Kingsmore at Rady Children's. And, that one was over 24 hours, I think, just under 24 hours at the time. And so I'm just thinking in my head, as fast as the sequencing runs have gotten and as simple as this prep is and as easy as it is to do, I can absolutely see why somebody who wants to understand the full landscape of a genome for a rare disease, of which there are, between 6,000 and 7,000 different rare diseases, this is the enabling technology. Right. And they just that community hasn't had this tool yet. So that's really, really powerful.
Yeah. And as I said, it really is the beginning of the story or the wave. And you brought up a really important point. Because we're so engaged with the ecosystem, our customers, hospital systems that people are using, TruPath in particular, the evolution and the maturation of that technology is going to be accelerated. And this is really sort of the new mandate of Illumina right now is bring that customer in earlier, listen, how we improve our technologies. And it's really paying off.
Well, I think it goes back to something you said upon your reflections, right? Which is we're all in service of using technology to open up this information for the market, right? For researchers, for clinicians and others. And so that partnership is key. One of the other innovations that I'm really, really excited about and want to pick your brain about some is all the things that Illumina is doing, in fact, beyond the genome. Even if we're known as, the genomics company, you've actually been investing for quite a while in R&D on things that are not just the genome. So you want to bring us along on how you got there, what the thinking was, and where we're at right now?
Yeah, this has been a subject and personal passion of mine since the very beginning. And I think it goes back to my grad school where I dabbled in proteomics a little bit. Right now, over the last 3 or 4 years, we've launched a number of products and invested in a number of technologies to complete that biological information level. So you're absolutely right. We were known as the genomic company and mainly focused on DNA and RNA. But now we've got proteomics. Now we have our 5-base genome, which is solidifying our position in essentially epigenomics. So when you look at that biological stack of the information, the biological information, we've kind of checked the boxes of each of those ohms. Now, there's still a long ways to go in each of those, but at least we have a portfolio that allows you to interrogate that biological information in completeness. And this is what science is missing right now, right? 10 years ago, you couldn't do this. And once again, I go back down to that catch-22. If the researchers and the healthcare professionals don't have the right tool, then they can't fulfill their mission or they can't think of the next experiment. With our latest project, with the acquisition of SomaLogic, our Illumina protein prep, we're at about 40 to 50% of the whole proteome, circulating proteome, that you can interrogate at scale at a reasonable price. So that is the very beginning of this innovation wave. That's another one where we're just going to be improving that technology, where interrogating the circulating plasma proteome is going to be commonplace in a decade. It's going to be, everyone is doing that. It's just a normal technology.
So that will be almost as routine as we would like to think maybe
exactly
the whole genome sequencing is right now.
Yep, that's the vision.
So can you help explain why it's so useful for those multiple, like that full stack of biological information to be read out, together? Why can't I just, go get my mass spec data and then, my sequence and, why is it helpful to have all this together?
Yeah, the analogy I love to use, and it's probably because I studied the liberal arts way back in college, is, there's a genre of painting called pointillism. So, and it's basically pictures created from little dots. And the more you focus in, you can't see anything, right? But the more you focus out and you have more colors, and the colors are basically the levels of biological information. As you focus out and you have those colors in the picture, you start to understand what the picture is about. But if you only are at the very focused level of only looking at one color and one dot or one piece of information, DNA or RNA, you can't understand the whole picture. And this is what we mean about filling in the biological comprehensiveness across the biological information, and then, of course, the multimodal. So that's single cell. Of course, we've been doing bulk forever. So single cell and then spatial is the next frontier too. And then looking at that organization across that information against the multimodal, across the whole genome level. Now, of course, the other axis we're not even talking about is across time.
That's right.
So as you can imagine, I like to think about, once again, we're at another big inflection point. Mm-hmm. The tools are becoming available, all powered by the readout power of our NovaSeq X. Mm-hmm. None of this could be viable without that power of being able to read out biological information based on sequencing at scale for a viable cost. So all of this is based on that. And that's the real key that's been unlocking all of this power. So it's absolutely a key point here, right? But where is this going in the future, right? So it's amazing to think about. Library preps and preps that actually can perturb whole biological systems like the CRISPR technology is allowing us to understand at a comprehensive system level changes or characterizing what's going on. But you couple that with the power of being able to read out that biology, all centered on one instrument, the NovaSeq X, and then you tie that into the abilities we have at Illumina here with AI. What we're building is a digital understanding of the world from the lab. That is an inflection point in science right now, and it's all powered by these innovations that are coming together. So when we talk about the next wave, that is what we're building here at a very high level.
That is very cool. And one image that was coming to my mind is almost, you're reminding me of like Illumina's Syrah, right? But you're painting these beautiful pictures over the course of time. And the X is your platform. We have covered so much ground on innovation, and I'm a little bit surprised we haven't actually talked about the box, right? I think historically that's what people associate Illumina innovation with. And so it was maybe a bit contrarian, or I've heard customers even say it's revolutionary that at the last Advances of Genome Biology and Technology, AGBT meeting, we didn't announce a new box. Can you tell us what the thinking is there?
Yeah, so that was intentional. And it really has to do with understanding our customers and where the market is today. So 2 decades ago, it was a novel technology-driven marketplace. To be honest, we could launch any new technology and people would just buy it and eat it up. Today, the major change over the last 2 decades is people want stability. Sequencing is a routine tool now in the lab, and people are buying our instruments to run businesses.
Good point.
They're buying our instruments to run clinical labs. So the innovation needs to be in another vector. Designing in the roadmap, and this is why those roadmaps are so important, the NovaSeq X, we plan for an upgrade, which we announced at AGBT this year, of actually producing 40% more output, new flow cells, the 5B, new sequencing kits. So what this does is it increases the value of people who own the NovaSeq X today. So that's compounding its value. And that's much appreciated. And from listening to our customers and what people want, that was a conscious decision to do that.
And I that brings us all the way back to the beginning where we started, right? Which is this process, this innovation process that you've really built into the foundation of this company, of thinking about the future, of thinking about how to unlock the next enabling technology for our customers, working with them. And then now we're riding this third wave with them. Sounds like predicated on the X and the ideas that you've been bringing forward from within R&D for kind of the last few years, even before they saw the light of day.
Yeah, so at a very high level, this is the beauty and the magic of Illumina and its innovation process. So it is just a privilege to be part of it. And I wake up every morning with the same amount of passion I had with the day I started it. So it's just an incredible environment around here. It's just thrilling to come to work each day.
It is. And I feel privileged that you shared this time with us on our very first video podcast. I don't think we could have had, a better guest or conversation as we really understand those who are driving this revolution. So thank you so much for your time, Steve. I really appreciate it.
Thank you for the time.
Thank you. Thank you all so much for joining us for this very first episode of The Sequence, and we hope to see you next time.